Hi everyone. Today we want to look at the role of vitamin A in vision. Uh, Severally, we have had people saying uh, people who have deficiency in vitamin A, they experience what we call night blindness. And therefore, we want to see exactly how is vitamin A related, or what exactly does it help in vision. First of all, we can look at the classification of vitamins in general, and we are saying vitamins can be classified into two. We have what we call fat-soluble vitamins. We have water-soluble vitamins. So the fat-soluble vitamins, we have vitamin A, vitamin D, E, and K. So what people call ADEK. And then the water-soluble vitamins are what we call the B complexes. The B complexes are vitamin B1, vitamin B2, B3, B5, B6, 7, 9, 12, and vitamin C. So this is what we call the water-soluble vitamins. But our interest today is to look at vitamin A. So what are the sources of vitamin A? One, we can get vitamin A from animal products, and we can also get vitamin A from plant products. When you get vitamin A from animal products, they come in the form of retinol. But when it comes to plant products, they come in form of carotenoids. And therefore, after consumption of food from plants, these carotenoids must be converted into retinol before absorption. So when you look at the animal products, you can have milk, eggs, liver, and fish. When it comes to plant products, you can have carrots, you can have sweet potatoes, pumpkins, and spinach. So what you need to remember is that in plants, vitamin A is in form of carotenoids, and therefore it has to be converted into retinol in the intestines before absorption. But when you take it from animal products, it is direct in retinol form, and therefore it's absorbed as retinol in that matter. So how exactly is vitamin A transported from the stomach and up to the eye? Because we want to see how exactly vitamin A reaches the eye. And what happens is, in the diet, after consuming food, food goes into the stomach and then into the intestine, where digestion takes place. After digestion, vitamin is absorbed. If it was in plants, it goes conversion to retinol. But if it's obtained from animal products, it's directly in retinol form, and therefore it's absorbed. After absorption, it gets into the circulation. In the circulation, it circulates for quite some time, and then it goes into the liver for storage. In times of need in the eye, there is a protein called retinol binding protein, which binds to retinol in the liver to form what we call retinol binding protein complex. And it is this complex that now transports retinol, that is our vitamin A, into the retinol pigmented epithelium in the eyes. This RPE retinal pigmented epithelium is a layer that is found on the structure of the eye called the retina. And in the retinal pigmented epithelium, vitamin A is converted into retinol 11-cis retinol molecule. Then this molecule goes to what we call the photoreceptors in the outer segment of a photoreceptor. So you need to remember that this retinol, what we call 11-cis retinal, will get into the outer segment of the photoreceptor, and it will combine with a protein known as opsin. After combining with the opsin, it now forms rhodopsin. So we are interested in rhodopsin because that is the photopigment that we want to look at in the rods, because its function is to absorb light. So these are the structures of the rods, and this is the cone. These are what we call the photopigments, and they are found in the retina. So what you need to know is that the rhodopsin that we are talking about, the photopigment, is stored in the outer segment of these photopigments, photoreceptors. So from this point here, it shows the outer segment. And then what you need to remember is that there's a slight difference between rods and cones in terms of photopigments. The photopigment in the rods is rhodopsin, while the photopigment in the cones is photopsins. Another fact you need to know about rods and cones is that we have a ratio. The ratio of cones to rods is about 1 to 2 to 20. This means in every one cone, there are about 20 rods. Another thing you need to remember about these photoreceptors is that the rods are sensitive to dim light. The cones are sensitive to bright light. So we have seen what stores our pigment is the discs, and the discs are found in the outer segment of the photoreceptors. And you have seen that there are two types of photoreceptors. We have the rods and the cones. We have also seen that the rods are sensitive to dim light, while the cones are sensitive to bright light. Another thing you need to understand is that the cones also have what we call subtypes. We have 3 subtypes of cones. We have those that we call, those that are sensitive to red wavelength. We have those that are sensitive to green wavelength. And then we have those that are sensitive to blue wavelength. If you look at these colors, you realize that these are what we call the primary colors. So all other colors originate from these 3. You need also to remember that the red wavelength is the longest, the green wavelength is the medium, and then the blue wavelength is the shortest. This one is about 565 nanometers. This one is about 535 nanometers. And then this one is about 540 nanometers. So functions of rhodopsin, we are saying it's a photopigment that absorbs light to initiate the process of phototransduction. And what is phototransduction? That is just conversion of light energy into electrical signal, what also we can call impulses or an electrical potential. What you need to understand is that energy cannot be lost, but can only be transformed from one form to another. In this case, we want to transform light energy to what we call electrical energy, because the nervous system understands only one language, and that is the electrical energy. And before you get onto the electrical energy, it means our photopigments will have to undergo what we call photochemistry. Then from there, that is when we will be able to get what we call phototransduction. In photochemistry, we are using light. This is just chemical reactions after exposure to light. So this is the steps of photochemistry. One, when light hits the rhodopsin. The rhodopsin, we said, remember, it's a molecule that has two parts. We said it has what we call the opsin, and then it has what we call 11-cis. 11-cis retinal. When light hits rhodopsin, this molecule here, what we are calling the 11-cis retinal, is converted into what we call all trans retinal. That is the first thing. The 11-cis retinal is converted to all trans retinal after light hits the rhodopsin. So what happens is, once this is converted into all trans retinal, it causes a conformational change on this protein we call opsin. And the first change is that this opsin will be transformed into what we call beta-rhodopsin. What we are indicating with B. Then from there, it will now be converted to what we call limo-rhodopsin. Then from there, we will convert limo-rhodopsin to meta-rhodopsin 1, what we are referring to as M1. Then M1 will be converted into meta-rhodopsin 2, what we are referring to as M2. That is the first stage. The M2, the meta-rhodopsin 2, activates transducing by converting. There is a hormone, there's a protein called G. It's called G protein transducing. So this meta-rhodopsin 2 will convert this G protein transducing into another conformational change. What you need to know is that this protein has 3 subunits, what we call the alpha, the beta, and the gamma subunits. This G protein transducing has 3 subunits. So on these subunits, on the alpha subunit, there is an attachment of what we refer to as GDP. And GDP is just guanosine diphosphate. So what happens is that metauxin will convert the G protein transduction, and this GDP will be converted into GTP. Mark the difference between D here and T here. This is guanosine diphosphate and this is guanosine triphosphate. So what happens is that once the GTP is activated, it will now dissociate. Once it dissociates, it will activate an enzyme called PDE. PDE is what we call phosphodiesterase enzyme, and the function of this enzyme is to break down what we call cyclic GMP. Once it breaks cyclic GMP, it will cause closure. It will cause closure of sodium and calcium channels. Once we close the sodium and calcium channels, it means the cell will be hyperpolarized. It will be hyperpolarized. And that's why we are saying from PDE, when it breaks down cyclic GMP, it will cause the sodium and calcium channels to close. And when they close, the photoreceptor cell will become hyperpolarized. It means if, for example, the resting potential of the photoreceptor was negative 40 MV, MV is just millivolts. What we mean by hyperpolarization is that it will become more negative. Maybe it will shift to minus 60 MV. So the next question is, what is the effect of hyperpolarization? When you have hyperpolarization, this amplitude will change the potential, of the membrane, what we are calling the resting potential, and it will lead to some cascades that will cause the signals that are then picked and transported to the brain. In the primary visual cortex, what we are referring as Brodmann area 17, where interpretation of the signals is done, and then you are able to visualize. So particularly what happens, you can see here, this is an example of the eye. All that we have explained takes place in the retina. That is where we have the photoreceptors, and after transduction, the optic nerve takes the impulses to the brain for interpretation. Thank you.